Good afternoon, I'm Jim MacAllister. I'm an evolution geographer from the University of Massachusetts, and I'm going to be speaking on a new view of evolution. It's just a brief outline of my talk. We're going to see a little synopsis of the view that has held sway for the last 70 years, called the modern synthesis, or sometimes referred to as neodarwinism. Then there's going to be this little thing called a debate in a nutshell, just to highlight the fact that the folks who are neo-Darwinists are very exclusive about their ideas. I'll define symbiosis and then talk about the new symbiotic biology. And then I would talk a little bit about a new view of evolution, which is symbiogenetics, which is symbiosis over evolutionary time, and Gaia Theory. And then I'll finish up with the question: how will a new view of evolution inform and affect geography and other disciplines? So I'm going to show a clip from the 2014 series Cosmos: A Space-Time Odyssey, and the host, Neil deGrasse Tyson, does a very nice job of giving us a synopsis of the modern synthesis.
If life has a sanctuary, it's here in the nucleus, which contains our DNA, the ancient scripture of our genetic code. DNA is a molecule shaped like a long, twisted ladder, or double helix. The rungs of the ladder are made of 4 different kinds of smaller molecules. These are the letters of the genetic alphabet. Particular arrangements of those letters spell out the instructions for all living things, telling them how to grow, move, digest, sense the environment, heal, reproduce. The DNA message, handed down from cell to cell and from generation to generation, is copied with extreme care. A specialized protein proofreads to make sure that only the right letters are accepted so that the DNA is accurately copied. But occasionally, a proofreading error slips through, making a small random change in the genetic instructions. A mutation has occurred. A random event as tiny as this one can have consequences on a far grander scale. That mutation altered the gene that controls fur color. It will affect the production of dark pigment in the fur of the bear's offspring. Most mutations are harmless. Some are deadly. But a few, purely by chance, can give an organism a critical advantage over the competition. A year has passed. Our bear is now a mother. And as a result of that mutation, one of her 2 cubs was born with a white coat. When the cubs get old enough to venture out on their own, which bear is more likely to be able to sneak up on unsuspecting prey? The brown bear can be seen against the snow a mile away. The white bear prospers and passes on its own particular set of genes. This happens repeatedly. Over succeeding generations, the gene for white fur spreads through the entire population of Arctic bears. The gene for dark fur loses out in the competition for survival.
So that is the modern synthesis and population genetics. And I'll just go back over some of the things that Neil deGrasse Tyson talked about. First, the modern synthesis is a big like us zoological model. It's basically modeled on animals. It treats the nucleus as a sanctuary. The genome is isolated from the environment. The genome is the ancient book of life. It's nearly immutable, but occasionally an error slips through. A random in relation to function mutation occurs, or an accumulation of these mutations occur, and this is what gives us new traits. He also said that most mutations are harmless. That's true because most mutations do nothing. They're buffered out. The system of the cell has many ways of dealing with errors in copying, and so the consequence of most mutations is that they make no difference whatsoever. If they weren't buffered out, almost all of them would be deleterious or fatal. The individual with the new trait is the unit of selection. It is only the individual that is naturally selected. There is no group selection. Then sex spreads the trait through populations. There's the survival of the fittest, or the fact that in this case the white fur is so much better at camouflaging that it gives you a selective advantage, means that the brown bears that live in the Arctic don't survive. And when the accumulation of different mutations has advanced far enough, you get reproductive isolation from the rest of the population, so that you have a new species, the biological definition of speciation, where the branch, the white bears, can no longer interbreed with the original brown bear population and produce fertile offspring. Here's the only diagram that Darwin included in On the Origin of Species. Darwin was illustrating what he called the principle of divergence, and you can see that it's a tree diagram with all the branches diverging. They fork or bifurcate. Now there's a little bit of conflation that goes on in the modern synthesis. People knew that offspring did not always look like their parents. So some part of the germplasm was causing that trait. When Mendel came along with his genetics, we then had a gene, and a gene then equaled a thing that created a trait. And then with Crick and Watson, you had a gene being a length of DNA that encoded a protein. And these two definitions, a gene that gives you a trait and the DNA encodes a protein, as you saw, are conflated because, in fact, a single gene mutation almost never gives you a trait. So symbiosis and symbiogenesis are two ideas in biology that have had a long history, except that most of that history took place in Russia and in the Soviet Union. The symbiogeneticists who did this work did not think that the idea of natural selection, Darwin and Wallace's great idea, could explain the creation of new traits, because it was an elimination process. It took a wide swath of novelty and narrowed it down. So there's these two definitions. Symbiosis is kind of an ecological term. It's the physical contact between two or more differently named organisms for most of the life of at least one of the organisms, or symbionts as they're called. Symbiogenesis can be the evolutionary consequence of symbiosis, where the symbionts begin to become a composite organism. This may begin with changes in behavior that lead progressively to physical, metabolic, and then genomic changes. Symbiogenesis can be instantaneous or it can occur over geological time. And unlike random mutation, it has been documented in the lab, in the field, and in the fossil record. In 1969, Lynn Margulis published a paper called On the Origin of Mitotic Cells, and from that work, which posited that mitochondria and chloroplast, mitochondria, those things that produce ATP in the cell, chloroplast, the photosynthesizing organelles of the cell, that those both had once been free-living bacteria and had been incorporated in the cell as organelles. The reason it's called serial endosymbiosis, endosymbiosis being these things getting incorporated inside of, is that these gray triangles mark three different symbiotic events. The first, between swimming bacteria, spirochetes, and archaebacteria, produce the first protists, single-celled eukaryotes, or cells that have nuclei. Bacteria do not have a nucleus. The second is the incorporation of mitochondria that gave the early protists the ability to breathe oxygen. This was important because the cyanobacteria over here, who are photosynthesizers, were contaminating the atmosphere with their waste product of photosynthesis, which is oxygen. And oxygen was toxic to most things on Earth which were anaerobic at the time. From there, we get animals, plants, and fungi. And then the final endosymbiosis is the incorporation of the chloroplast, the incorporated cyanobacteria that produced algae and plants. Lynn Margulis' idea, this idea was first ridiculed, and then when the technology and the science came along that had convincing and overwhelming evidence of it, it was accepted but very begrudgingly, and it was claimed that it only happened way back in geologic time, or if it didn't happen only then, that it was of minor importance. So this is a little exchange between Richard Dawkins and Lynn Margulis at this 2009 Homage to Darwin debate at Oxford University. And Dawkins says, if you take the standard story for ordinary animals, and what's wrong with it? You've got a distribution of animals, you've got a promontory or an island or something, and so you end up with two distributions of animals. And Lynn asks him if this is a geographic example, and he says yes. Just geographical. And then on either side of this promontory, you get different selection pressures, and so this one starts to evolve this way, and that one starts to evolve that way. And what's wrong with that? It's highly plausible, it's economical, it's parsimonious. Why on earth would you want to drag symbiogenesis in when it's so unparsimonious and uneconomical? And Lynn answers, because it's there. So this is the kind of exclusivist notion that people have had about evolution. And the tipping point came, I would say, really in 2009 through 2013, first with James Shapiro's book, Evolution, A View from the 21st Century, which lays out the molecular biological evidence for the fact that symbiosis and symbiogenesis is much more common in evolution than was previously thought, and also lays out a lot of other things that basically counter the modern synthesis. Now, the things that Neil deGrasse Tyson talked about, that list that I showed you, all of those assumptions and rules have been broken. They're not all wrong, but they're either not applicable to most of the biota on the planet, they're exaggerated as far as what their importance is, and in some cases, they are just simply wrong. And then two papers came out, the Gilbert paper and the McFall-Nye paper, and these papers are basically manifestos of the new symbiotic biology. They're basically saying that the evidence is overwhelming and that a tipping point has been reached. And then finally, Dennis Noble, who's a professor of physiology and systems biology at Oxford, has his paper where he essentially shows that all of the rules and assumptions of the modern synthesis have been broken. I'm going to show you a few slides from Scott Gilbert's talk called Expanding Lynn's View, A New Symbiotic Biology. If you search on YouTube and just put in Expanding Lynn's View or Scott Gilbert, you'll get it. But I'll go through some of the parts of this. First, he talks about Lynn. He particularly likes her quote, "I don't consider my ideas controversial, I consider them right." And he thinks about her in the same way that he thinks about Einstein, in sort of general relativity and special relativity. Special relativity being her endosymbiosis idea, and her general relativity idea being universal symbiosis, or what I will refer to as Gaia Theory. Now, one of the things about the modern synthesis is the unit of selection. It is that little white bear cub, the individual, that is selected. And a very radical change in biology is that the entire notion of individual is being replaced. And there's a lot of ways that one could think about how one is an individual. You could be an anatomical individual. You know, our body is one genome that integrates cells to a common wholeness. You know, I know her, she knows me. That's that kind of notion. There's a physiological individuality. The integrated organ systems of your body are all working toward a common end. There's a developmental notion of individuality. We are all the products of the fertilized egg. The idea of immune individuality, that our self is protecting us, guarding us from the other, the non-self. There's this notion of genetic individuality, that the genome of each cell from the sperm-egg fusion is nearly identical. So even a heart cell, a liver cell, a skin cell, all contain the same genome. And then there's the idea of an evolutionary individuality, that the genome provides the phenotypes for natural selection, and that individuals are either organisms or, if with Richard Dawkins' sort of selfish gene notion, the genes are the things that are selected in individuals. The new symbiotic biology says, no, there's no real individuals. There is what's known as the holobiont. The holobiont being the animal plus its persistent microbial communities. We have over there, we have a cow. Lynn used to like to say that a cow was a 40-gallon fermentation tank on 4 legs. A cow ingests grass, but it cannot digest grass. It relies on its microbial community and its rumen to do that work for it. So what is a cow without its microbial community? It's a dead cow. Here we have coral, and the coral polyps, the animals that make coral, rely for their carbon resources on the zooanthellae, the algal symbionts that they have. And in fact, what coral bleaching is the loss of those symbionts, and without the symbiont, the coral does not have the carbon resources to survive and dies. Termites, which everyone hopes will not infest their house and ingest it, they ingest wood, but they do not digest wood. They rely on the microbial community of anaerobic protists and bacteria that live in their hindgut, and we have some of them here. And I'm just going to show you a little clip of one of them, Mixotricha paradoxa, which lives inside the termite. And here's Mixotricha paradoxa, which means mixed up pairs paradoxical, because it was thought that these were cilia, but in fact this protist is coated with about 4 different types of bacteria, 3 spirochetes and 1 rod-shaped bacteria. And the spirochetes, the little swimming bacteria, are the things that push it around. So this is an organism that's actually composed, we call it the poster child for symbiogenesis. It's composed of 5 different genomes. So what Scott says is that we are all lichens. Does everybody know what a lichen is? See them on trees over there. A lichen is an organism composed of a fungi and a photosynthetic partner. In this case, it's an alga, but it could be a cyanobacteria. And that we are all products of networks of symbiosis. And here's an orchid. The orchid has a fungal symbiont colonize it as it germinates to supply the embryo with its carbon source and vitamins until it's able to photosynthesize. The individual is derived from the same source, the fertilized egg. Well, the holobiont research says, no, that's not true. Over 90% of the cells in our body are prokaryotic. That means they're bacteria, they're not nucleated cells. Each of us has about 160 of these necessary symbionts, and the human race has about 1,100. And you have different ecosystems of these microbes. In your airway, on your skin, your right hand ecology differs from your left hand. For instance, the vagina, the intestine, the mouth all have their own ecosystems of bacteria. Genetic individuality, where all the cells of the body have the same nuclear genome, which are replicates of the genome established at fertilization. Well, the oocyte over here that's being created from these nurse cells is passing along this red material. And that red bacteria in this Drosophila is Wolbachia bacteria. It's actually being passed through the germline. So the Drosophila is actually born infected with the Wolbachia bacteria. Now, we don't get our bacteria passed down vertically that way. We get it horizontally. So as soon as the amnion breaks, the baby is exposed to the microbial community of the birth canal. The baby gets bacteria from nursing, from touching its parents. This is the way that infants get inoculated with the kinds of bacteria they will need. In fact, one of the things that's come along now is this idea of the hygiene hypothesis, which is that a lot of chronic illnesses, inflammatory bowel disease and others, are actually a result of children being too sterile and not getting the bacteria they actually need in their gut. And you may have heard of things that are now called fecal transplants, where they actually take the microbial community from a healthy person and put it into a person that doesn't have a healthy microbial community, just to basically to cure those kinds of problems. Now here are some aphids, and they're born red. If they're infected with Rickettsia, the aphid newborns turn green. So here you have a phenotypic trait. You have the color of the animal actually changing based on the microbe that infects it. In a different kind of aphid they've studied is infected with a bacteria called Buchnera, and the Buchnera will create a heat shock protein which the aphid will use then to be able to survive and breed at higher temperatures. Now there's a little bit of a trade-off. The amount of fecundity of the insect is less at high temperatures, but the fact is that it can still produce. So there's another way that the bacteria is actually changing the animal. Here's a parasitic wasp. It's going to inject its egg into this little aphid, and the egg will hatch into a larvae. The larvae will eat the inside of the aphid out, eat its cuticle, and then fly off as a wasp. Except that if the aphid happens to be infected with, in this case, H. defensa bacteria, it may very well be able to kill the egg inside it and survive. Now, the interesting thing here is that when I say may, it may because the bacteria that's infecting it actually has to be infected itself with a phage, because the defense against the parasitic wasp is actually bestowed by the phage of the bacteria. So you have the symbiont of the symbiont giving the animal a trait. Here's symbiont-produced variation in humans. Sushi-eating Japanese actually get a horizontally transferred protein from red algae that affects the type of strain of Bacteroides that they have in their gut. And the result of that is that they actually get much more nutrition out of their diet of raw seafood than Western populations do. Developmental individuality. The individual is an organism from ovum to ovum as progeny of the zygote. Environment may be permissive or detrimental to development, but it does not have an instructive agency. It doesn't help the animal along. Here is a solar-powered sea slug. It's a mollusk. And when it's born, it has a mouth and it eats the chloroplasts from Valcheria, a type of seaweed. And it eats the chloroplasts, but it doesn't digest them. Instead, it distributes them in the diverticula of its gut that you see as kind of that leafy structure, and then the rest of its life it basically lies around in the sun and lives off the photosynthate that the algal symbiont manufactures for it. So I guess that would be the environment helping you out. Here's the development in another one of these parasitic wasps. And what you have over there is a stained preparation. You can see here's the nucleus, and here's Wolbachia bacteria, close-up of the nucleus, close-up of the Wolbachia. And then in some of these, what the researchers have done is they've treated them with rifampicin, which is an antibiotic, and it kills the Wolbachia. And what happens to the wasp? Rather than having a healthy reproductive system, the reproductive system is completely withered because the wasp relies on the bacteria in order to form its reproductive tract. And the way that this happens is that without the Wolbachia, apoptosis or programmed cell death kind of takes over where it shouldn't, and that's what basically causes the reproductive tract to wither. Another example, over here we have two pictures. On the top left, the villi of a germ-free mouse, and on the bottom, the villi of a conventionally raised mouse. You'll see the germ-free mouse's intestine lining has poorly developed vasculature because the microbial community produces the metabolites required for the development of the vessels in the villi. And the vasculature in your intestine is actually dependent upon the microbiota in your gut. Bacteria modulate the expression of genes involved in important intestinal functions, including nutrient absorption, mucosal barrier fortification, xenobiotic metabolism. That's if you eat something very strange, how it gets metabolized. Angiogenesis, the formation of blood vessels, and postnatal intestinal maturation. And right now there's over 100 bacterial species in the gut that are being researched to see how we outsource various developmental phenomena. This study, which has this quote, there's a diagram from the study, colipase, which is involved in lipid transport. The difference here is between a germ-free mouse and a mouse with its normal gut bacteria. Colipase, the conventional mouse, has 6-fold more colipase in its bloodstream. And the second thing, angiogenin. Angiogenin is that metabolite that promotes the growth of vessels. The conventional mouse has 10-fold more. And the other thing about angiogenin is that it kills Listeria bacteria, which the mouse doesn't want and you don't either. And you rely on your symbiotic bacteria for angiogenin 4. And SPRR2A helps form the lining of your gut. And here it's a 50-fold increase that the conventional mouse has over the germ-free mouse. Eupremna is a little squid, and it basically is born without a light organ, but when it gets to be an adult, it has a light organ. And it actually collects Vibrio fischeri bacteria on its skin, and the bacteria actually create and populate the light organ. The light organ actually casts a bioluminescent light that masks the shadow of a squid, because without that light organ, it would cast a shadow and predators would know that it was swimming in the water column. So here an entire organ of an animal is being created by bacteria. So normal gut microbiota modulates normal brain development and behavior. During evolution, the colonization of gut microbiota has become integrated into the programming of brain development, affecting motor control and anxiety-like behavior. So, for instance, the neurotransmitters that you have in your bloodstream, serotonin, is not manufactured by you. It's manufactured by your microbiota. We're not individuals in a developmental sense because bacteria are involved in all of these different kinds of systems. Things like obesity, etc., are controlled by your microbiota.